lobe is called caudate lobe. Another way to know which one is quadrate, which one is caudate is quadrate lobe is just next to the gallbladder. So this is the gallbladder. Quadrate is just next to the gallbladder. Okay, make sense? Do you remember inferior vena cava? It's a big vein, right? So inferior vena cava is actually attached here. That's why you see a nice, depression here for inferior vena cava. And that is the sulcus for inferior vena cava. Next to the caudate lobe of the liver. So, that is the liver. If you cut the liver and see inside, under the microscope, the liver tissue, beautifully structured. You see, this is how under the microscope you will see many hexagonal lobules. Hexa means what? 6, right? Penta means what? Okay. So hexagonal, that means 6 angles in those lobules. Okay. So beautiful. And if you see one lobule, hexagonal 6, okay, 6 angles, and in the center of the lobule you have a canal that is for the central vein. So this canal is for central vein. And then from the central canal, liver cells, these are called hepatocytes. So inside the lobule, you have the liver cells, hepatocytes are present. And they beautifully line up like this. Okay? So these are hepatocyte means cell, hepatocytes. Okay? Hepato means liver, cyte means cell. Okay. Now, this is also a lobule. This is the central vein. Here, central vein. And these are hepatocytes. These cells, see individual cells, hepatocytes. And at each of those 6 angles, you have portal triads. In each angle, you have portal triad. This is one portal triad, this is another, that is another, okay? Like this. And in each portal triad, you have 3 things. Bile duct, portal venule, that means vein, small vein, and portal arteriole, small artery. Make sense? So, a venule, an arteriole, and a bile duct. 3 things. In each portal triad, you have those 3 things. Okay? Arteriole, venule, and bile duct. Same thing in each portal triad. So this is portal triad, which contains bile duct, arteriole, venule. Now you see one thing. Beside each hepatocyte, you have a very thin hair-like, bile canaliculus singular. So, bile canaliculi attached attached to each cell. You see how beautifully, right? Done. And they collect the secretion from each hepatocyte and bring it into the bile duct here. Make sense? So those are tiny branches of bile duct. They get the secretion from each cell, right? And then give it to the larger bile ducts, okay? In each corner. So that's how the structure of the liver, microscopic structure of the liver. And the bile is secreted from the hepatocytes. So hepatocytes secrete bile, and then the bile enters into the duct. So it is like this. These are hepatocytes, and you have tiny canaliculus. So secretion enters into the canaliculi and then goes to the portal tracts, the bile duct or the portal tracts. Okay. Now, the functions of hepatocytes, the cells of the liver. The hepatocytes process the nutrients. How many of you remember after absorption of nutrients, I told you that nutrients go where? To the liver. Because first processing of nutrients occur in the liver. Remember that? So, processing of nutrients, number one. Number two, store of fat-soluble vitamin, vitamin A, D, E, K, fat-soluble vitamins. A, D, E, K are water-soluble vitamins. Fat-solubles are the other vitamins. Then perform detoxification. That is another important function of liver. Detoxifies the toxic chemicals that are produced in the body or enters from outside. Like, you know, when you take medicines, most of the medicines have also chemicals that can harm your body. You know that, right? That's why we say adverse effect, right, of medicines. So there are good components, but also there are chemicals that can cause harm. So those chemicals must be detoxified, neutralized inside the body, right? And most of detoxification occur in the liver. And that's why you will see in many medicines it says, if you have liver disease, talk to your doctor before you take this medicine. Okay? Because if you have problem in the liver, then it will not be able to probably detoxify that chemical, right? That can accumulate in the body. So, liver detoxifies the toxic elements. Production of bile, about a liter, 1 liter of bile, like 1000 milliliter. It says here 900. Some books will say 100, 1000. That's fine. So, about a liter of bile is produced by the liver per day. Okay, and then this diluted bile, 1 liter, right? 1 day. Liver produces how much? About a liter. That's a lot, right? So that will go to the gallbladder. But gallbladder is small, right? What happens? Most of the water from that diluted bile will be absorbed, taken back into the body, right? So gallbladder concentrates the bile, okay? Takes the water out, highly concentrated, okay? The bile. And it stores it. So liver produces or secrete bile, and gallbladder concentrates and stores bile. Bile. Bile is a yellow-green alkaline solution or secretion that contains bile salt. bilirubin. Those two things. Because of bilirubin, the color is yellowish. Because bilirubin is an yellow colored pigment. Yellow, right? So that's why the bile is yellowish. Now, bile salt is very important because bile salt, I mentioned, right? Bile help in fat digestion. Remember that? A few minutes ago, I mentioned that pancreatic juice help in digestion of everything, right? And bile specifically help in fat digestion. So, bile salt does what? You see, these are large food pieces, okay? Here, bile enters through the bile duct, and the bile salt molecules get attached to the food molecules. Here, the outer surface of the food. So now let's see here. This is the food molecule, and these are bile salt molecules. They get attached to the surface of the food and will act as detergent. You know, you do laundry, the detergent you use, they do what? The detergent molecules do what? They get attached to the dirt, surface of the dirt, okay? And reduces the surface tension. It reduces what? The tension of dirt, okay? Particles. And will break the dirt particle into many tiny pieces, okay? So it will get detached from the clothes. Make sense? So the bile salt molecules act exactly as what? Detergents. They will get attached to the surface of the food and will reduce the surface area. So when the stomach will move, or the intestinal wall will move, not stomach, intestinal wall will move, then it will easily get into multiple tiny pieces, breakdown. So that is the digestion, right? Breakdown of large to small. So that's the digestion of fat. Okay, of fat. Act as a detergent and break the food into small pieces. Further, fat. Large intestine. If you see the parts, you have cecum, which is the first part of large intestine. Then you have colon. Now colon has ascending colon, transverse, descending, sigmoid. So ascending colon, transverse colon, descending colon, sigmoid colon. Okay? So those are the parts of the colon. Then you have rectum and anal canal. Okay? So this is the cecum, very first part of large intestine, then ascending, transverse, descending, and this is sigmoid. Okay? And then you have rectum and anal canal. Okay? So those are the parts of your large intestine. This is a tiny structure attached to the cecum. This is called what?
Appendix.
Appendix. Vermiform, like worm-like structure attached to the cecum. That's why it is called vermiform or worm-like appendix. I have talked about this probably before. Appendicitis is a problem, right? This appendix has no use in our body. Basically, no use. There are some lymphoid follicles, but you know in the wall of intestine you have aggregated lymphoid follicles, right? Called the Peyer's patches. We don't need those small amount of lymphoid follicles. Make sense? So basically, appendix has no function in your body. Scientists believe that this is an anomaly. That means probably maybe a million years ago, this structure was bigger, much bigger in the human body. Okay? Gradually got smaller and now it's staying like this small. Okay? So, so the removal of appendix is called the inflammation of the appendix is called appendicitis, and appendicitis could be dangerous. You see, when infection occurs in the appendix, swelling occurs, right? And inside the appendix, you have the fecal materials, right? The food stuff, feces, and when swelling occurs, if you don't remove it, it may get bigger, 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 and rupture, burst, right? That will cause the release of fecal materials in the abdominal cavity. It will spread everywhere, right? That could infect other organs, okay? And that can cause sepsis and infect other organs and poisoning. It's like poison inside your abdominal cavity. That could be dangerous, okay? Yeah. So, now you see, the large intestine has sacs, pouches, or pocket-like structures. Those sacs are called haustra. Haustra is plural, haustrum is singular. Haustrum. One is haustrum. If I indicate many, haustra. Okay? Those sacs. And another structure, you see, a band of muscle is attached to the wall of the large intestine. That is called tinea coli. You need to know all these things. Haustra are those sacs, and tinea coli is a band of concentrated muscle fibers attached to the large intestine. Another thing, you see, these are fat structures, yellow, fat or adipose tissue structures attached to the tinea coli, and these are called epiploic appendages. Epiploic appendages. Fats attached to the tinea coli.
Wait, so where are those haustras then? Where are they? I thought they were the yellow things. Oh, I gotcha.
These sacs.
Okay.
Okay. So those are few things that you can see from outside in the large intestine. Those sacs are called what? Haustra. And the thick band of concentrated muscle fibers, tinea coli, and those fat structures attached to the tinea coli are called epiploic appendages. Okay? Make sense? So that is the large intestine, and this is mesentery. We have talked about mesentery, that hangs the GI tract to the wall of the abdomen. So greater omentum. How many of you remember from the greater curvature of the stomach? You remember I said drapes down. So that covers the front of the abdominal cavity, right? So that is the greater omentum. They have done what? Greater omentum covers like this here. They have moved it up like this. It's like, tablecloth hanging like this. You can just move it up like this. Okay, so they have done it like to show inner structures under. Okay. Rectum. Rectum is a part of large intestine, and anal canal is the last part of the large intestine. And in anal canal, you have two sphincters. This is important to know that in the last part, canal, you have two sphincters. One is here, and this is the outer one. So this is internal sphincter, and this is external anal sphincter. Okay. What happens when you defecate? Defecation occurs. First you know that this is skeletal muscle. Skeletal muscle. And this is smooth muscle. So, two sphincters are formed by two different types of muscle. Okay? Internal one is what? Smooth. And the external one, skeletal. Make sense? Which one is voluntary, which one is involuntary? Skeletal is what? Voluntary, right? All these are skeletal muscle. Voluntary. Smooth is? Involuntary. So which one we can open if we desire? This one, right? This is voluntary. This is involuntary. So what happens? First, we voluntarily relax the external anal sphincter because we can control this. So we voluntarily relax this. And then when we do that, a signal goes to the spinal cord. When you relax it, the signal goes to the spinal cord and tells the spinal cord that external one is already open. So spinal cord will do what? Send signal to the internal one to relax it, because we cannot do that. Spinal cord will send signal to internal to relax it. So when both are relaxed, the recess gets out. So that's the mechanism. Okay, so it's interesting. Like, you know, one is voluntary, another is involuntary. First you open the voluntary, then signal goes to the spinal cord, then comes down to the internal, okay, sphincter. Okay. Pancreas, the last thing, okay. So pancreas is this one, you remember? Now, I told you that pancreatic juice enters into the duodenum. Remember that? Has protease, amylase, lipase. They help in digestion of the food, right? Digestion of protein, digestion of carbohydrate, digestion of lipids. Is it clear? So pancreatic juice is very important to digest the food. But inside the pancreas, you also have some structures. Those are called pancreatic islets. Pancreatic islets. Pancreatic islets produce insulin and glucagon, not pancreatic juice. Pancreatic islets produce two important hormones, insulin and what? Glucagon. Insulin does what? Lowers or increase the blood sugar? Lowers. Very good. Right? That's why diabetic patients take insulin, right? To lower the blood sugar. And glucagon does just opposite. So glucagon does what? Increases the blood sugar. Make sense? And by the action of these two, your blood sugar is regulated. Now you tell me, if you take a lot of food after you take heavy meal, food, your blood sugar goes up, right? So which one should be secreted? Insulin, to lower the blood sugar, right? Keep it in normal range. Now if you are starving, you didn't take food a couple of days, which one should be secreted? To elevate the sugar, right? So by the action of these two, our blood sugar is controlled. Now if the islet cells are destroyed, one type of islet cells are called beta cells. Beta cells, if the beta cells are destroyed, beta cells secrete insulin. So insulin secretion will be stopped. Make sense? So that causes type 1 diabetes. Okay? If pancreatic beta cells are destroyed, no insulin will be secreted or less insulin will be secreted. Make sense? So blood sugar will tend to stay high. That is diabetes mellitus. Okay? So just know that pancreatic islets produce insulin and glucagon, and those are responsible for regulation of what?
Blood sugar.
Blood sugar. And other cells, those are called pancreatic acyni. Yeah, very good. Acyni. Pancreatic acyni cells produce pancreatic juice. Okay. So, yeah. So you have the islets and acyni.
It's not a C, but it does sound like Nagini. Nagini. I remember that.
Does this make sense with the juices?
Yeah. It's got pancreas.